Well, I'm excited to be here among all these young and talented scientists. Not if I'm not young, I'm just saying. But it's fantastic to be here. It's my first time at Gatsby in summer school. And it's congratulations to all of you for being here. That's amazing. It's a huge achievement, and you should be all proud of yourself. That's amazing. So I'm Mojgan Rabiey. I'm an assistant professor at University of Warwick. And in my group, we work on phage or bacteriophage. I'm going to tell you about it, all about it. But so from this slide, you can see I'm going to talk about brassicas. I'm going to talk about cherry. I'm going to talk about phage cocktail 9 a.m. in the morning. But yeah, so I'm going to tell you a little bit about how we create a phage cocktail in the lab, and how we use that to control plant diseases. So let me tell you a little bit about myself. Now at Warwick, I have my own group, and we just work on bacteriophages to control important crop diseases that I'm going to tell you a little bit about it. Right, so one of the pathogen systems that we work in the lab is called black rot disease in brassicas. And we all here know about brassicas: cabbage, kale, broccoli, cauliflower. We consume them every year, right? Or every day, basically. And as a nation, we produce around 300,000 tons of vegetable brassicas each year. That is worth around £400 million to the UK economy. So it's huge for us. But if you look at these images here, so you could see that symptoms of black rot disease on brassicas. So you could see this V-shape, almost symptoms on the leaves. This is how this pathogen enters through the leaf. But this is caused by Xanthomonas campestri, pathovar campestri, or from now on I call it XCC. So when it happens to a farm, farmers usually have to leave their products behind. Why? Because the only control method is that they have to pick up all these infected plants one by one. And in a huge farm, it's almost impossible, right? It's time consuming, it's labor intensive, it's expensive. So then farmers have to just leave that, abandon their field. So it's very costly for the farmers because this is their livelihood. So, and we can't use antibiotics anymore. So we used to use copper or streptomycin, but because of the antibacterial resistance, because of the environmental impact, but also because of legislations, we can't use antibiotics anymore. But you might say, oh, why don't we just use resistant cultivar? This is because this XCC, or Xanthomonas campestri, is a complex pathogen. And by complex, I mean these. You could see here we have around 9 races of XCC, and within each race we have different strains. So this is based on, like, we at Warwick, as part of the Xanthomonas threat consortium, they sequenced around 500 different isolates of XCC, and they found that they are very genetically diverse. So even if we say, okay, we have a resistant cultivar of brassicas against race 1, what about race 4, or what about other races? So it's difficult to breed for resistance as well. So from this phylogenetic tree, you could see that race 1 and race 4, we have loads of strains in the UK, and race 1 and race 4 are the most dominant races in the UK. So then it is important that we find a solution to tackle this important crop disease that is very complex, is very diverse, and is almost impossible to treat using chemicals or resistant cultivars. So now I'm going to talk about phage, my favorite microorganisms. So phage are viruses they are natural viruses that specifically infect and kill bacteria. So these are one of the most abundant microorganisms on Earth. So they are everywhere. Everywhere you look, say in soil, in leaf, in bark, in water, in rivers, seas, in human guts. And I was so pleased to see a research article in BBC a few days ago that they talked about the phage in human guts and how they're going to use that to treat important diseases such as Pseudomonas aeruginosa that cause lung infection, or like Staphylococcus aureus that cause like infection on the skin and so on. But so how a bacteriophage infect a bacterial cells? So they bind or attach to the bacterial cell surface using receptors that I'm going to talk later. They inject the genomic material inside. They take over or hijack the replication machinery of the bacterial cells. They make their own particles and bang, they lyse and burst the cells open. And each bacteriophage goes and infects a neighboring cells. And this is the beauty of using bacteriophage, because there's a live interaction between phage and bacteria, or we call it arms race dynamic. But it's not like antibiotics, that if we use antibiotics and bacteria resist to that, we need to find another antibiotics. But for phage, it's a live interaction. So there's always like phages that infect bacterial cells using the receptors. And even if bacteria resist somehow to the bacteriophage, phages evolve to overcome that resistance. So this basically interaction goes on and on until like the phages keep the population of bacteria under control, under the threshold that they can cause disease or damage. So I told you about receptors on bacterial cell surface. So on gram-negative bacteria like XCC that I told you about, you can see that we have this outer membrane here, and on this outer membrane we have a series of receptors, but the bacteria have them all on their cell surfaces for many different reasons. For example, we have pili here. That is usually bacteria use that for attachment to surfaces or like making biofilm formation and so on. We have flagella that bacteria use that to move around, so that helps with the motility of bacterial cells. We have lipopolysaccharide. That's again is on the outer membrane, and it's very important for virulence and pathogenicity in bacterial cells. And we have a series of purines and so on. But bacteriophages could use this receptors on the bacterial cell surface and to bind and then initiate the infection. But bacteria also try to resist, right? So they don't want to get infected, they don't want to be killed, right? So what they could do, they could, for example, mask this phage receptor, so phages cannot bind to this, or they could modify the cell surface receptors, so then this attachment site on the phage cannot work anymore. Okay? But then bacteriophages find a way. They also evolve this attachment sites, and they find a way to bind to the bacterial cell surface again. But also this is important, that when we want to use phage and find phages, we should find phage that uses different receptors. So if one phage uses flagella as a receptor and one uses lipopolysaccharide as a receptor, then even if resistant image and if bacterial cells mask one of those receptors, then there's other phage that could basically come and infect using the other. So there's always ways that bacteriophages could interact with bacterial cells. But another thing that we need to consider when we're talking about phage-bacterial interaction is anti-phage defense system. So through evolution of phages and bacteria for years and years, bacteria developed this system we call anti-phage defense system. So it's very complex. I'm not going to want you to understand everything like in one go, and because this is a complex image. But I just want to, for example, mention that for example, bacteria has developed this, for example, the method to degrade phage DNA. We have CRISPR-Cas, like a molecular scissor that basically, again, helps to interfere with phage infection. We have enzymes and chemicals that helps to stop phage replications, and we have suicide. Bacteria make suicide, so they kill themselves, so phages cannot replicate, and so on. But this is so pathogen to pathogen is different. So we can't say like XCC has this antiphage defense system, because it's complex. There are different races that they have different antiphage defense systems. We also have like Pseudomonas that uses different systems. So it's not as easy to say if we look at the genome and they have this antiphage defense system, we know what phage to use. I'm going to tell you a little bit about this later. But now we know all about this, so you know about phage receptors, you know how phage bacteria resist to phage, you know about antiphage defense system. So now I'm going to tell you about this phage and XCC interaction and what we do in the lab, basically. So one of the best parts of the job is we do a lot of field sampling. So for example, we went to this brassica field last summer. You could see how beautiful and colorful. And students were taking samples, and I was just taking pictures. I promise I did some help, and look how happy I am, having free food for dinner, right? And it was the most delicious cabbage I ever had, fresh from the field. But yeah, so then we took the samples to the lab, and we do like a basic plaque assay. And what it means that we mix the samples that we collected from the field, we do serial dilutions, and we do plaque assay. So you could see these plaques here. So each of them is like a one phage infected one bacteria cells, then produced all those little phages, and they infected their neighboring cells, and that's why you could see these clear zones. But you could see each one, like each plaque has different sizes. So what we do then, we purify each plaque that has different size. For example, this one that is quite a pinhead size, to this one is massive, and we purify this more, and we end up with like around 50 different phages. And then the first thing we do, we genome sequence these phages. For example, here I'm showing you one of the phages, 2.2, that we collected, and we do electron microscopy because we can't just see phages by naked eye. We use a transmission electron microscopy, and you can see that this phage has a beautiful head and a very long tail. These are among cyphoviruses. So based on these genome sequences and looking at the shape and morphology, we narrow the phages down to 3 phages. And one of the first things we do when we collect phage, we do a lot of killing curve assay. And what I mean by that is that we measure the bacteria population, or we call it optical density, over time. So here I'm showing you like 24 hours reading of a bacteria population absorbance. Here you could see the pathogen on its own, so you could see over time it grows exponentially. But when we add phage, so we have 3 phages here, phage 1.5, 2.2, or 4.3. When we add phage, you could see that XCC population cannot grow. So immediately it kills the bacteria population, and bacteria population stays down. And you can see that by the end of like 24 hours, we don't see any bacteria-resistant image. So if bacteria-resistant image, we would have seen like, yes, bacteriophage kills the bacteria population to begin with, but then resistance would have emerged like here. But even if we continue this killing curve assay for 72 hours, we could still see the bacteria population is down. So as I said, at Warwick, we are very lucky. We have like collection of 1,000 XCC isolate. So what we did, we used like around 200 of those XCC isolate, and we tested all these 3 phages against to see which phage can kill what race and what isolates. So here dark color means that phages can kill the bacteria population very nicely and clearly and effectively. Light green means that somehow they kill, but not as effective as the green one, the dark green. And white, it means they cannot kill the bacteria population. So you could see that, but you can't, because these are a lot of isolates named, you can't see the numbers. But basically, these bacteriophages are very host specific. And this is the beauty of using phage again, because they are very host specific, they only use the receptors that there's specifically on the XCC races, and they cannot infect any other microbes. So if we have a phage that infects XCC race 1, it cannot kill other XCCs, or it cannot kill Pseudomonas, or it cannot kill any beneficial microbes. So then it's like safe to use them as a biocontrol. So as I said, so the phages are host specific to race 1 and race 4. And as I mentioned before, these races are the most dominant races in the UK. So Then, but how do we design a phage cocktail? So I told you about, like, yes, we want phages that they use different receptors. We want phages that basically target anti-phage defense system and and we want phage, when we want to use this phage cocktail, we want to target as many strains of XCC as possible within each race. Oh, and this is what happens when I use Mac to create images. So it's a bit, but you need to trust me in this one. So here we test the first thing we do when we want to create phage cocktail. We test different concentration of phages. We want to see what concentration is best to use or titer to best to use. So they kill the bacterial population as effectively and as quickly, but no phage resistant emerge. So we found what concentration to use. For example, here we have this concentration that kills the bacterial population and we don't see any resistance. And what we do next, we need to understand the receptors on the phage, on the XCC surfaces. So we do what we call resistant experiments when we use a very high concentration of phage to kill the population of XCC immediately. And when the resistant bacteria emerge, we collect those resistant isolate and we sequence them. And when we sequence those, then when we have the sequence back, we check them against the reference genome. That's the XCC, for example, here. This is this strain 8978, and we find the mutations. And where the mutations happens, those are the ones that normally used for receptors. For example, this phage 1.5 uses lipopolysaccharide to bind to the bacterial cell surface, but for example, this phage uses flagellum. So now we were quite lucky to find phages that they target different receptors. So very important for making a phage cocktail. So, and I told you about anti-phage defense system. So here, the anti-phage defense system, the presence is in blue color and absence in white. And you can see that almost each strains or isolate, they have different anti-phage defense system. So that's why I said it's very complex. So we can't say because they are within one race, they should all have the same anti-phage defense system. But what we see here, there's a pattern. So those here, the phages can infect this the first part, and you could see that they lack this anti-phage defense system, but here they are almost present. But we need to do loads and loads of different, test different isolates. So as I said, we have around 1,000 isolates in-house, and we are doing more analysis to understand whether there is a correlation between anti-phage defense system and how phages infect bacteria cells, because that would be like very cool if you find there's a correlation, because then we could just look at the genome and say, oh, this phage could infect this antiphage, this bacteria cell, because it doesn't have this antiphage, or it does have. Right, so now we want to make the phage cocktail. So what we do, then we try to mix phages together. So we have combination of 2 of phage and combination of 3, and we again test this by measuring the bacteria population over time. We have XCC on its own, but when we added phage into that, whether they are single phage or combination of 2 or 3, you can see all of them could significantly reduce bacteria population and effectively. So this, then we found, okay, so we could basically use all these 3, the combination of 3 of these phages as a cocktail or mixture, and then we could use them to treat XCC. So then we do a lot of implanter experiments, and what I mean by that, we infect the brassica leaves with XCC and with XCC and phage, and we try to look at the population. So one of the group in Warwick, they luminescently labeled XCC, race 4, and here as I said, so XCC enters through the edges, and you could see 7-day post-infection. The symptoms of XCC could be image them, and you could see that they spread. And by 14 days post-infection, you could see that it spread all around the leaves to the stem and to the veins on the stem. But when we add phage to this to XCC, so 7 days post-infection and 14 days post-infection, you can see that we almost have no XCC on the leaves. So this is very promising for us. but we need to do more and more experiments to understand this. For example, this is like we call pricking method, when we force pathogen to enter through the leaves. But this is not as simple as that in the environment. So if bacteria cells could infect leaves through different methods. For example, they could enter through the stomata and so on, or through any wounds and so on. So what we're trying now at the moment to do, we try to use different methods. So we're trying to use a spray method, because that will be easier for farmers to use. We're trying to do a seed coating method, that we infect the seeds with phage and pathogen and understand where the phages happens. But another thing that we try to do in the lab, we are trying to label our phages with green fluorescent protein, or GFP. So then we have a luminescent labeled bacteria, and then if we have a labeled phage, we could easily see what happens, where do the phages go. So this is one of the unknown. When we apply phage to the plant, we don't know what happens after. Do they stay on the leaves? Do they go systemic? Do they go to the vein? Do they go to the stem? So that's the thing we still need to find out. And that's why by labeling the phages with GFP, that's because, but labeling phages with GFP is quite challenging, because for bacterial genome, we know a lot about them. We know almost all the genes' functions, well, not all, but almost. But with the phage genome, half of the genes, we don't know what the functions are, basically. So that's quite difficult to understand, okay, what genes we should target or what to do. But at the moment, we know, so for example, for XCC phages, we found out some of the function of each genes. I'm not going to go into that details, but and then we're trying to understand, okay, where we could put this GFP label that could help us with because when we also put the GFP in, we don't want that impact the phage replication and so on. So that's the next step for us. So we're trying different methods to infect, but also trying to label our phages. So far so good. We have a nice story for XCC so far. So happy brassicas for us. So now I'm going to go to talking a little bit about different pathosystems. So I'm going to talk about cherry canker. So vegetable brassicas for us is an easy system to work on, because we could basically grow brassicas in the lab, and we have like growth chambers that we could basically do a lot of experiments. But working with trees is different, it's challenging, it's just because trees, basically when we want to work with trees, it has to be in the glass house. We can't just have them easily in the lab, right? But also we have to have a right season for that. We can't force the trees, for example, trees to flower for us at the right time that we want. So when they go to flower, we have 2 weeks or 3 weeks window to do a lot of experiments on the flowers. And then for the leaf, we have only 2 or 3 months to do all the experiments. For the bark of the tree, if we want to do infect the woody tissues, then we have to wait until winter time, and we have 1 month period to work on those. So it's very challenging and different systems to work with, but it's actually fun. Right, so cherry canker, right. So this is caused by Pseudomonas stringi. That's another pathosystem, and again is a complex disease like XCC. So Pseudomonas stringi, Pathoa stringi, and Pathoa muruspunorum, we have different races. And I put this phylogeneity here because I wanted you to see that, for example, PSS, Pseudomonas stringi, Pathoa stringi, or PSMs, they all belong to different phylogroups. And what it means that they have different pathogenicity and different virulence. And that's why it makes them difficult or complex. Right, so, and this is a pathogen that is around all year round, right? So it could infect the leaves. You could see these shot holes on the leaf. You could see that it infects the fruit, so necrosis on the fruit, and huge amount of oozing and bleeding from the trunk of the tree. It seems that trees almost bleed to death. So, and for the farmers, this is the livelihood. The only method that they could control this at the moment is just to cut down the trees. And it's not like brassicas to say, okay, so next year I'm going to plant brassica again. But for trees, it's going to take years for the farmers to plant them again, and until to the point that they produce fruits. So basically, so UK also produces around 25 million pound worth of cherries each year that we export quite a lot of that. So again, it's important for the UK economy. And again, there's no control method. We can't use antibiotics anymore because of the reasons I explained. And again, we can't just say, let's use a resistant cultivar, because this pathogen is, again, is very complex. There are different pathovars, and within each pathovars, we have loads of different strains. So I'm not going to go into detail. So again, so we went to cherry orchards, we found phages, we did all this characterization, we genome sequenced, we do a lot of killing curve assay, plant assays, and from the genome sequence we found 70 phages, and we genome sequenced them, and based on genome sequencing we narrowed the phages down to 5 phages, and I call these MR phages. They come from my name, Mojgan Rabiey. So it's cool to have phages on my name, and also these phages belong to very unique, all belong to different families, and one of the families is called Mojo viruses, from my name, Mojgan. So it's good, like you see all my phages are on my name, so it's very cool to do that. And then what we do here, again, we do test them against loads of Pseudomonas stringi, and again, we found they have different host range, they could infect, but again, they cannot infect any of the beneficial microbes like Pseudomonas fluorescens. They are very host specific. And so you know, for XTC phages, I said they use different receptors. One use flagella, one use lipopolysaccharide. But this MR phages, you use mainly lipopolysaccharide as their receptors, but they use different genes. So different genes within the gene cluster. So for example, some of these genes, and how do we confirm that they are the right genes for the receptors? We do a lot of like gene deletion. So what we do, we find for example, these 3 phages, these 3 genes that I mentioned here, we target the genes, we delete that part of the gene from the gene cluster, for example, in terms of glycosyl transferase within the lipopolysaccharide gene cluster. And this is the exact genes that we are interested in. This is again within the outer membrane. We delete this gene using the method that we have in-house. And what we do, we then repeat the killing curve assay. And here that phage could kill the wild type PSS, but when we deleted the gene, phage cannot infect this anymore. So then it shows that this is a target receptor. We deleted loads of genes, so around more than 10 genes, and we found these 3 genes are the one that they are specifically phages use as a receptors. Then again, so we do look at the anti-phage defense system, and for PSS, we don't see, even for XTC, we saw some correlation, but here we don't see any. We know the anti-phage defense system are very the phylogroup dependent, but then there is no correlation between anti-phage defense system and phage infection profile. So, but these are only we found, like we looked at 250 strains. Again, we need to do more analysis to understand specifically what it means by anti-phage defense system. So then we do a lot of like infection assay in the glass house. So it has to be in the right time. So usually we do them in winter. We have 1 month window to do the experiments. So we infected the cherry woody tissues with the PSS and each MR phages, like 5 of them, and the cocktail of 5, a mixture of 5. We measured like disease score. We look at the bacterial population count. And if you look here, we have the pathogen only. So you can see the brown lesions. We have, for example, 1 MR6 was a single phage. You can see they reduce the symptoms, but when we use the cocktail of mixture of 5 phages, you can see that almost clear the symptoms. So we compare to the negative control. So it shows that this phage cocktail is working very effectively when we infect the cherry woody tissues. And when we measure the bacteria population, colony forming unit, here you can see on the y-axis, and you can see PSS and phages together. You can see that phages significantly reduce bacteria population. So we do a lot of these experiments. We do them in woody tissues and leaves and so on. And after all these experiments, we say, okay, we need to do a phage field experiment. So in the UK, we don't have any commercial phage products. So you could see that we can't just easily go and do field work or field experiments. So these phages I'm talking about, these are natural viruses that we collected them from southwest England, and we haven't done any like genetic modifications or anything. But still, if we want to use them in the field, we have to have license, and we can't just go and release a phage, although they come from the environment. We can't put them back. So we had all the paperwork, we had the license from DEFRA, and then this is the first ever phage field experiments in the UK. So we had the license, we went to our experimental plot, and we went to the field not knowing what to expect, because it's the first time we're doing the field work. We don't know how long whether phages could survive after we apply them. If they survive, how long they would survive. Would the phage resistance emerge in bacterial population in nature? And if phages impact microbial communities? So basically loads of questions we had in mind when we did this field experiment. Right, so we went to this cherry field. We selected our cherry trees that we wanted to work with. We collected leaf and shoot samples to begin with. We wanted to make sure there is no natural or native phage and PSS. And then after that, we infected the cherry leaves and shoot with pathogen first, and then with phage after. And we collected samples every day for 4 days, and then after 30 days. So we had different treatments. So we have buffer only, I call PBS. We have phage cocktail. We have pathogen only, PSS, and we have pathogen and phage. And from all the samples, whether we applied phage or not, we collected we re-isolated the PSS population using a specific media. We did a lot of PCR to make sure they are the one that we put in. And we also re-isolated the phages we put in. And we did loads of experiments on those to understand this phage-bacterial interaction in the environment. So what I'm showing you here, I'm showing you the phage count. So we call it plaque forming unit or PFU. So it's a phage count only. On the top you have the baseline. So we have the time point. We have baseline before we started the experiment, and then then every day for 4 days, and then day 30 after we started the experiment, after the infection. So you could see that baseline, basically we don't have any phage. That's good. So we have a buffer, we have phage cocktail, we have PSS and cocktail together. So that's good that we don't have any phage to begin with. Then we infected the trees with bacteria and then with phage, and we collected samples. And when we look, you can see here, when we have the phage cocktail on its own with no pathogen, after 3 days we couldn't detect the phages anymore. But when we have the host, basically bacteria, because the phages use host as replication, so they rely on the host to replicate. So you could see that we could still detect phages in day 30. So it's a huge finding for us, because when we go to farmers and we talk about phage, and these farmers that we go to, they are very open-minded. They are so excited and interested to learn about science, to learn what we do, and they're open-minded. They always let us take samples from their farm and field. And when we talk about phage, the first question they ask us, okay, so what happens to the phage when the pathogen is not there anymore in terms of safety? And so we know that, so if pathogen is not there, so we saw that after 3 days we couldn't find phages, but if the pathogen is there, yes. But also we collected samples at more time points, so at day 60 and day 90, and then we couldn't see any phages after. So it means that if we apply phage into the field, we need to reapply maybe every month if we wanted to keep the population down. Considering that in the field everything is different, so for example in the lab and in the glass house we have everything under control. We know the temperature, we know the humidity, we know everything. But in the field everything, so temperature from morning to evening is different, humidity is different, we have rain, we have wind, we have UV is different from what we do in the lab. So it's huge to understand this, but we need to do more of this field work. We need to basically collect data to show how safe phages are to use as a biocontrol. Okay, so then I said, okay, we're also interested to understand whether phage resistant emerge in the field. So what we did, so as I said, we re-isolated the pathogen we put in, and we challenged them again with wild-type phages, and wild-type phages could reduce the bacterial population. It means that resistant didn't emerge in the bacterial population, because if we had resistant, wild-type phages wouldn't be able to kill. So again, so in nature it's quite different what we see in the lab. In the lab we use like rich nutrient media. We basically provide everything for the pathogen to be a pathogen, but in the field it's quite different. And to understand the impact of phage on microbial community, we did like 16S and ITS sequencing. These are the ones that we target basically different regions, like 16S in terms of bacterial population. And here what I'm showing you is the alpha diversity from day 3 and day 30 samples. And if you look here, time has the most significant impact on the bacterial community. And when we did statistical analysis, we found that phages, they didn't impact any other microbes. So because they are very host specific, they only impact PSS, so they didn't impact any other bacterial community, but also fungal community. When we targeted ITS region, we did lots of sequencing, and again, so day 3 and day 30, time has the most important impact on the fungal community. So it was a very productive and very good field work for us. It was very labor intensive. It was here, so you could see how many places we only use in one day of work. So we made this phage, but it's a huge teamwork, and everything basically we do is not just from one person. It's like we work together to make this happen. So what is the practical use of phage? How do we use phage in the field if we wanted to use? So we could use them as cure the symptoms. For example, you could see as injections. So make the systemic go up and down the trees. We could use them as a spray method. We could use them as an irrigation system. We could use them as a seed coating. But all these experiments need not only we just do them in the lab, we need to do them in the real world environment, so we could understand the phage-bacterial interaction in real time. So what else we do in my lab? So we try to understand this phage-bacterial interaction in detail. So what we're trying to do, we're doing a lot of metabolomic and transcriptomic experiments. So we try to extract lots of metabolite from trees when we infect them with phage and pathogens, and to understand, so we do a lot of RNA sequencing to understand the gene expression, and we want to understand whether phage could boost the defense system in plants, for example, or if plants could remember phages and then they have this defense system always activated. As I said, we are labeling our phages in XCC and Pseudomonas with GFE so we could track the movement of phage in the plant environment. Also, we try to understand how phage shape the evolution of pathogens, for example, in terms of antiphage defense system and receptors and so on. So in that note, as I said, it's a teamwork. Loads of students have been in my lab, for example, undergraduate students, master students, PhD students. So last year I had students that were supported by VSP. So I think one of you asked yesterday, like, what other societies support summer studentships? So for example, BSPP, British Society for Plant Pathology. They have, it's a very generous society that they have funding for undergrad summer studentships. They offer 15 projects almost every year. So these are the projects that supervisors apply for, and then they are from different universities across the UK. We have Royal Society of Biology. For example, 2 of my summer students at the moment in the lab, one of them is supported by Royal Society of Biology, and one of them is supported by University of Warwick summer studentship. So usually, like, these societies open their call for funding early in the year, because then if you want to start in summer, so you have to start looking early. So usually February, March is the time that they open the call, and then you could go to the website and apply for the funding that you want. But they also, these societies are, as I said, they are very generous. They provide travel grant. So if you want to go to a conference, for example, during your time as an undergrad, they are very generously support students to go to conferences. So you could become a member. It's quite a reasonable price, and they support you throughout your study to go to conferences. But also they have outreach fund. For example, BSPP has some outreach funds. So if you are interested to talk about plant science and you want to do some outreach activities, you want to go to schools and talk about this, but you need some materials, these societies also provide outreach funds so that you could apply for and basically get that started. These are the funding bodies that supported the experiments, and as I said, this is a teamwork that lots of students been involved, and it wouldn't be possible basically to do all without the efforts of the students. And in that note, thank you very much for listening, and happy to answer.